Hi, thank you so much for having me. Today, I'll be talking a little bit about what our lab's been working on a project titled about arbitrium-mediated crosstalk. And just to preface all of this, we all know that communication is really important across all walks. That includes us yapping about really cool phage science here today. That includes other high, quote-unquote, complex eukaryotes having communication, as we've learned earlier today about quorum sensing in prokaryotes. And of course, why we're all gathered here today in phages as well. And the reason why communication in phages is super important to study is because phages don't live in solidarity. They live in these complex microbiota type communities where there's a bunch of other bacterial hosts, phages in tandem. And within this community, there are a group of phages, temperate phages, and as we know, they can alternate between either the lytic cycle, where they replicate, lyse, and eventually kill the bacterial host, or the lysogenic cycle, where they integrate in the host chromosome. And how temperate phages can alternate between these life cycles is dependent on a lot of factors: chemicals, stressors, as well as arbitrium, which is a small peptide communication system you find in phages that infect Bacillus species. They control the lysis-lysogeny decisions, as well as induction based on phage population density. And they gauge this phage population density based on levels of a signal peptide called AMP. Basically, when there's low levels of AMP in the environment, that's a signal to the phages that there's relatively fewer numbers of them out there. And so arbitrium favors the lytic cycle so that the phages replicate, kill the host, and take advantage of the abundant host resources out there. When the AMP signaling peptide levels are high, that's a signal that there's relatively high levels of phages out there. And so arbitrium instead guides towards the lysogenic cycle, where you integrate into the host chromosome and so preserve the host for future propagation. Something to keep in mind is even as a lysogen, the phage continues to produce this AMP signaling peptide. Besides the signaling peptide, there are also two other key players in the arbitrium system. There's AIM-R, which is the peptide receptor, as well as AIM-X, an effector gene that guides lysis, the lytic cycle. Key message though, more AMP peptide, more lysogeny. Less peptide, more lysis. And why do we care about arbitrium? Well, it's ubiquitous in Bacillus in a lot of genomic and environmental contexts. You can find them in phages that infect probiotic species, tidal sediments, mammalian gut, soil, and even in pathogens. Something to also know is that besides phages, you can also find arbitrium systems inside conjugative plasmids. Most notably, the virulence plasmid PXO2 in Bacillus anthracis. And we've learned yesterday how Bacillus anthracis is pretty scary for a variety of reasons. And also, just to further show the diversity of arbitrium systems, this is a phylogenetic tree of the different arbitrium systems out there, and they're grouped based on the receptor AIM-R's homology. And at the moment, my project focuses on this branch right here of sp beta-like phages. Zooming into this little branch, you can see that all of the phages in this little branch has their own arbitrium systems with their own peptides that recognize their own receptors. In this family, the peptides, the signaling peptides, are made of 6 amino acids. And what's really cool is that the last 3 amino acids are recognized by identical residues by their own respective AMR receptors. And instead, what confers binding specificity are the first 3 amino acids in those signaling peptides. And so what that tells us is that there's a very tight evolutionary space where the phages have to evolve to get that arbitrium signaling specificity. And all prior structural studies have claimed that arbitrium systems have evolved to avoid crosstalk in a sense that because where they each bind to their own receptors. But because of this tight evolutionary space, we did ask, is it possible to have crosstalk where one peptide could bind and be recognized by a receptor of another system? And that would be really cool because that would unlock or show us that phages are capable of complex ecological behaviors, where, for example, here we have a blue phage that's about to infect, but if it senses and crosstalks with a lysogen of peptide, it might actually be forced into, for example, lysogeny. So to answer this, first off, our collaborators from the Alberto Marina lab in Valencia have conducted some thermal fluorescence, where they took the AIM-R receptors of 3 different phages, ATCC 13952, GO11, and SP beta. They took these AIM-R receptors, they mixed them with candidate AMP signaling peptides, and they measured their binding stability. The key takeaway message with these graphs is the further the curves are along to the right, the more stable the binding. And as you can see, first off with ATCC, besides its own peptide, the one in blue, it could bind to one other non-cognate peptide. GO11 could do the same thing with one other peptide. And for SP beta, there are 2 other relatively stably binding peptides. And to China, I want you to draw your attention towards ATCC and GO11 because it seems that they could bind to each other's peptides in these thermal fluoresceins. And to take this a step further, Alberto and gang have also visualized and crystallized the structures for these 2 AIM-R receptors and bound them to not only its cognate AMP peptide, but also to the non-cognate seemingly working peptide, and have found that the conformational changes in this AIM-R receptor are the same regardless of the cognate and non-cognate peptide. So in vitro, crosstalk is possible. What about in vivo? And that's where my PhD jumps in. First thing I wanted to look at in terms of crosstalk in vivo was from the perspective of induction. And as a quick reminder, as I mentioned just now, when there is low levels of AMP, the phage is more lytic. So when a lysogen is stressed, it will excise, produce a bunch of phage particles, and you get to visualize a lot of phages. But if there's a bunch of AMP that is sensed, the phage is now more lysogenic, and the induction is suppressed instead. So with this concept in mind, I took those lysogens of those phages we saw earlier. I added a candidate panel of non-cognate synthetic peptides to them, and then I induced them and titered them and counted how many plaques formed. And the hypothesis then is, if the AMPs work, we should see less phages. Starting with SP beta, we can see that only peptide 2, its own peptide, seemed to have suppressed induction, which goes to show that even though we do see these in vitro stable binding, it might not reflect what's happening in vivo. However, with ATCC, besides its own cognate peptide, number 4, we did see lowered suppressed induction with another peptide, one belonging to GO11, the one that matches our in vitro data. And what's really cool to see is with GO11, besides its own cognate peptide, the non-cognate peptide from ATCC also seems to have suppressed induction. And so the takeaway message here is SP beta only recognized its own peptide for induction. But for GO11 and ATCC, they recognized each other. However, one caveat with this experiment is that I added these synthetic peptides at 5 micromolar, which is well beyond what we would expect of native expression levels of a peptide like that. And so I asked, what about lower AMP concentrations? Will we still observe these phenotypic differences? And so I took GO11, the one that had the biggest phenotypic differences, and I repeated this induction experiment with the cognate, the non-cognate peptide that seemed to work, as well as a non-cognate negative control. And what we can see here is that starting at these concentrations, 0.1, 0.25 micromolar, there seemed to be a difference in sensitivity between the cognate and non-cognate AMP that worked. And so that was a signal to us that it was important for us to continue these induction experiments at natural AMP concentrations. Because if a phage were to produce AMP at, say, 0.5 micromolar, great, there's crosstalk. But if it was at a lower concentration, that means crosstalk isn't naturally occurring. And it's hard for us to gauge the levels and concentrations of AMP in the media. So what we did instead was we took the phages that seemingly crosstalk with each other, and we swapped their AMPs using a CRISPR-Cas-based vector. Specifically, I took GO11. I first made a deletion of the 6 amino acids of the functional signal peptide, replaced it with ATCC, this peptide, the one that's supposed to work, as well as with 2 other peptides from phages that we didn't observe crosstalk in. And I took these swapped chimeric phages, and I did some induction experiments. First off, what I saw was, compared between the wild type and the AMP deletion had a much higher phage titer, And that's to be expected because without AMP, the phage is more lytic, more plaques. However, if you swap it out with the ATCC's peptide, you can see that the titers are restored back to wild type. And you don't observe this with the other phages signaling peptides. So that shows us only ATCC's AMP peptide suppress phage titers to wild type levels. And there is potential sensing of ATCC's peptide by GO11 at native concentrations. Besides that, we were also interested in, besides induction, the infection side of things, because that's also an important aspect of a phage's life cycle, where if you infect a bacterial population with phages without the AMP, you should see suppressed bacterial growth because the phage is more lytic. But with AMP there, the phage is more lysogenic, and you should see a restoration of the bacterial growth. And so I took those GO11 swaps, and I did these infection experiments against a bunch of naive cells. And what we could see was that between wild type and delta AMP, the AMP deletion mutant, we see that by deleting AMP, bacterial growth is worse. Makes sense. Phage is more lytic. But if you swap it out with ATCC, the peptide that works, you can see that bacterial growth is restored to that of wild types. And that's only for ATCC, not with the other signaling peptides. And so to summarize, synthetic peptide screening does show symmetrical crosstalk between GO11 and ATCC. They sense each other in induction. But there is a divergence in cognate, non-cognate AMP effect depending on concentrations. Having said that, GO11 does sense ATCC's AMP at native concentrations in both inductions and infection contexts, as we saw with the chimeric phages. In the future, I will be proceeding to make more of these chimeric phages with the other phages, as well as to, once I have these chimeric phages, possibly test the ecological consequences of this crosstalk. So that's about it for me. Thank you so much to Jose and Cora for supervising me, as well as to the rest of the Tenares lab for guiding me. And also shout out to Alberto and Nuria for a lot of their guidance and help and collaboration. But yeah, I'm happy to take any questions.